Good morning, everyone. Good evening, wherever you are. My name is Diana Wall, and I'm the science chair for the Global Soil Biodiversity. And I'm really pleased today to have you all here. You just saw the picture of Dublin, and we're looking forward to seeing as many of you as possible at our meeting in Dublin in March of 2023. And you can find out more information about that on the Global Soil Biodiversity website, I should say. It's a real pleasure to tell you what we're going to do today. We're going to start off with just a little housekeeping for all of you that are joining us from around the world. It's going to be a real privilege to hear from Patrick. But first, I have some warnings. We're recording this, so it will go on YouTube eventually, and you may want to use it in your classes or to get just your friends to hear more about the fantastic experiments that Patrick has done and what he has discovered around the world, looking at everything from macroarthropods to even nematodes, as we say. I'm very happy about that. Also, I just want you to know that the talk will go about an hour. And then after that, we're going to have questions, and you should put all your questions into the Q&A box. Don't use the chat, use the Q&A. That's going to be where all the questions will be. And I don't want to waste much of Patrick's time, but I do want to say this is the very first time we have had a GSBI lecture. Generally, we have held for the past year, and you may know about our webinars, where we bring together scientists interested in all aspects of soil biodiversity, whether it's biogeochemistry or whether it's molecular microbes or nematodes, what they're looking at, or soil science. And today, I'm very pleased to introduce my friend and colleague, Patrick Lavelle. Patrick?
Hello, good morning. Good morning, everybody. Well, thanks a lot for inviting me to this GSBI conference. I'm very glad to present. I think with Diana, we've had a very long cooperation, and it's always been very important for me being associated with Diana's initiatives. So today is just part of this long story, and I would like today to speak about soil macroinvertebrate. We have just published a few months ago a big synthesis on macroinvertebrate communities, and I think this is what I'm going to present today. Please confirm that you can see and hear me and see the slides.
It looks great, Patrick.
Okay, thank you very much. So I'm going to start. Macroinvertebrates. Well, okay. So we will define the soil macroinvertebrate, macrofauna, as everything that can be, any invertebrate can be seen at the naked eye. So it makes it quite simple to define. And you will see that it's a very highly diverse community. You know, in any site where you spend sometimes sampling, you will find 16 orders, more than 50 families, up to 400 species, you see. And I always complain that our children that know all the African mammal fauna, you know, they know zebras, lions, and hyenas, and this kind of things, but they know very little of just what they have under their feet, you see. So actually, you're really, it's amazing what you have. And not only in terms of diversity, but also in functional diversity, you see. We have these three groups that we used to call soil ecosystem engineers, earthworms, termites, ants, because they are the ones that really build and maintain the conditions, good living conditions in soils, you see. We also have these other groups that we call the litter transformers, who are the ones who compost all the organic matter that come to the soil, you see, diplopoda, isopoda, and many other groups. We have also predators, chilopoda, arachnida, and part of the ants are predators. And coleoptera, diptera that are mixed, that have mixed functions. Some of them are predators, others litter transformers, and also a few primary consumers. Although it's interesting to note that very few invertebrates eat living roots. So this is what we have, and so we can really say that we have three main adaptive strategies among this group: the ecosystem engineers, litter transformers, and predators. So what is living in soil? I think to really understand the importance of macroinvertebrates in soils, and not keeping on thinking that they are just here because over conditions have been created for them. We have to figure out what living in soil is. So imagine you are here. This is a kind of a thin soil section. You see, this is like a histological cut in the soil. This one has something like 5 centimeters in large and 10 centimeters high. You see, so you can see all the soil aggregates, macropores, and so imagine you're living here in this little hole. How do you feel? You see. Well, you may feel happy, you see. I say that because, you know, in English, you say something like the early bird catches the worm, so it's not very pleasant for the worm. But here where I'm living in Colombia, they say happy like a worm, feliz como un alambre. So I just start my serious conferences asking people why a worm can be happy. And you know, the worms have some very strange biological characteristics. First, they have 10 hearts, you see, so you can tell that they can have two love pains, and they still have 8 hearts to recover. And they also have 2 alternative sexes, you see. And it's quite an interesting story, because if you look really at what it is, you find out that these earthworms are obligate male homosexuals, but they have found a way to be fertile homosexuals, you see. So maybe they represent something in evolution that has to be really admired. No? Anyway, you know, this is why they can be happy, but there are also reasons not to be so happy, because when you live in soils, first, it's a very compact environment. So really, if you are not able to move the soil to dig, you cannot move. And one very important thing about soils is that microorganisms that move very little, if at all, because they cannot move, they very soon become inactive. And it's a very important feature in soil that bacteria, for example, that could multiply in 20 minutes in laboratory conditions. If you look at turnover times of microbial biomass, it's always several months to years, you see. So basically, microorganisms are most of the time inactive because they cannot move. The other thing is that you are in an amphibiotic medium. You're in this small hole, small pore in the soil. Now it's dry, but if it rains, it becomes totally filled with water. So you need to have a very specific respiratory apparatus to live in this condition. And maybe the worst thing in soil is the low quality of food, you see. You may think that soil has a lot of food, and it has, you see, you get lots of dead leaves and roots coming to the soil. But there is something strange about it, is that if you look at the histological cut in a dead leaf, you will see this black dot inside the cell. This black dot is a protein, phenol protein complex. What happens is that when the cell dies, the phenolic compounds that the plant used to fight against primary consumers, they are released in the cell content, and they will combine with the proteins that you have there, forming these phenol protein complexes that very, very few organisms can digest. Of course, we have in soil high quality resources like also that like the 10 to 20% root exudates that plants throw into the soil from photosynthesis. But they are released at very specific scales of time and space on the root tips. And if you're not present at this moment and at this place, you cannot use them. So basically, soil has so much restrictions for life that organisms have developed cooperation in their evolutionary story. You see. So I'm going to tell you a small story that you may have heard when you were a child, no? It is a Sleeping Beauty story. Actually, the Sleeping Beauty story was made by a soil ecologist, I guess. And in soil, you get the sleeping beauties, these bacteria that you can see here, you see. And you can see these bacteria in a dormant stage, surrounded by polysaccharides and with a clay particles blanket, you see. And you can see lots of these figures in soil, very thin sections, you see. And if it's about, if you speak of fungi, well, fungi don't have the same behavior, but they also, a lot of them resist as spores, you see. So you have all this microbial biomass sleeping, you see, and they may not be active for 1 or 2 or up to 3 years, you see. And they are waiting for the Prince Charming. The Prince Charming is an engineer, you see. In that case, an earthworm, but it may be an ant or a root, you see. And the kiss of the worm is the intestinal mucus of it. So after that, the story will not become so glamour, because the worm will ingest everything, you see, the whole colony with the bed and the blankets. And this is, well, a more scientific representation of the story, you see. We have the worm on one side that builds and organizes the soil. So when the worm ingests soil with the bacterial colonies that you see on the right, he will add in his in the foregut 1 volume of water to 1 volume of ingested soil, and 5 to 18% intestinal mucus. which is a very energetic glycoprotein. So what the worm is doing in the first part of the gut is mixing all these things in his gizzard. You see, earthworms have gizzards just like chicken. And in the first half of the earthworm gut, you get a culture medium, you see, which will allow bacteria to recuperate very fast, sometimes in less than half an hour. They will recover their enzymatic capabilities. And in the second half of the gut, mucus is totally reabsorbed by a process that we have no idea how it works. But there is no longer intestinal mucus there. It has been recuperated because it's a very high energy investment that the worm cannot lose. And microbes start to digest organic matter for the benefits of the worm and of themselves, because the digestion is external. So this system, this collaboration, this mutualism between microbes and earthworms can be seen with almost all ecosystem engineers. The same thing happens with roots, which put root exudates in the soil to activate microflora and get in turn nutrients released, and termites and ants also do some of it. So from this point, we have tried to build a general model of soil function that would take into account the creation of these interactions. Self-organization theory is a theory that will offer us the ability to put in a same conceptual model organisms of different sizes. You see, microbes, microfauna, mesofauna, macrofauna, and also all the physical elements that are very important in soil. Soil physics is very important because if you don't have pores, you don't have habitat. If you don't have connected pores, you don't have water infiltration and storage and so on. So what does this theory tell us? There is a very beautiful paper by Perry that was published in '95 that explains in very clear and simple terms a theory that when you go to the original books is very difficult to understand. So what Perry says is that self-organization is a universal property of natural systems. Chemists know it very well, physicians, and even sociologists use this basic conceptual background for their theory. A universal property in which interactions among biotic and abiotic components occur at discrete scales. So what it tells is that in a soil, at any small scale, all the organisms that are there will interact, you see. Well, maybe a root with a mycorrhiza or an earthworm with a bacteria. And they will create structures. The structures they will create, you will see that it's, in the case of worms and roots, it's macroaggregates. If it's ants, it will be more creation of pores. So they create structures, and these structures, the accumulation of these structures will change ecosystem function. We all know that soil aggregation and porosity is a very important attribute for things like plant production. There are two other properties of these systems. First, that they have a hierarchized structures, which means that we have structures at different scales that are embedded into the structure at the next scales. We will see examples of that. And they are metastable on the edge of chaos. It's a beautiful poetic expression. Metastable means that as soon as part of the organisms that create this interaction disappear, all the system disappears. So we have examples. It's something that to see that in soils needs really a very detailed observation of infection. But we have something here in Colombia that is typically one of these self-organization structures, you see. It's called the surales. It's mounds that are created in flooded savannas by interactions among plants and earthworms. The plants attract the worms with probably with the kind of litter they produce and possibly root exudate production. So the worms come there and they will deposit their cast at the soil surface. And by multiplying this process over time and sometimes thousands of years of casting all the time at the same place, you will get these very beautiful structures like that. And another example I like to use that is not so much related to soil is what is called flying rivers. Flying rivers is a name given for the transport of water from the Atlantic Ocean to the Amazonian forest and down to southern part of America by this process. So it's a very beautiful process. You see, it starts with evaporation of water in the ocean. Then the trade winds will push the clouds inside the Amazonia. Clouds will give rain. Rain will infiltrate into the soil. Here, trees will pump this water, 200 liters per day by a tree in Amazonia, 200 liters that go again in clouds that are transported further west by the trade winds, and from rain to cloud and soil, this water comes down to South America. So it's interesting to note that the same people who have a large crops of soybean in the Brazilian cerrados or in the Argentinian pampa, when they go to Amazonia and deforest Amazonia, they are just cutting their water supply. This is sometimes economic logic. No? Okay, so based on this principle, we have defined what we call soil functional domains. For example, one of these domains is a rhizosphere. It's a place in the soils where the roots are the main ecosystem engineer. And in this small picture of what a rhizosphere is, a very old picture coming from the previous works of David Coleman that I like to say hello today, and Clarholm. You see that the rhizosphere has interacting organisms. It has a frontier. I mean, it is operating at a small scale. It's creating structures, aggregates and pores, and all this has positive feedback for plant nutrition, and also for all the organisms that interact here, with a very important role of bacteria, microfauna, you see, amoebas that are there, and mycorrhizae. So soil is like a mosaic of such soil functional domain. You have the rhizospheres of the plant, you have the xylospheres of the earthworms, which can be defined by the sum of macroaggregates and pores created by earthworms, termitosphere, myrmecosphere for termites and ants, and something that Mike Beer and other co-workers have called aggregatosphere, that can be called simply the litter system. You see, all the decomposing part of the organic input where composting occurs. Okay, so now we have tried to put these functional domains in a global model that would also give place to other very important processes like food webs, you see. And I think in the elaboration of this model, there has been always a kind of a question on how to incorporate in this model the very important studies that have been done on food webs. You know that lots of people have worked on that. So it was important to get a combination of what we could call the ecosystem engineer approach and of the food chain approach. So this is how we represent it. We have structures created at different scales that are characterized by the organisms that dominate them and the structure that they create. So at the bottom, at a scale of a few microns, we have microorganisms in their microbial aggregates, the picture that we've seen. Then in intermediate aggregates of something like 50 microns, this is where micro food webs operate. Micro food webs are all these food webs that have been described by a number of researchers. Diana has been part of them, James Hunt, and many others that I should be able to name now. So what this micro food web is doing is approximate control on microbial communities. So it is the function of this predatory action that is very important to maintain stability and equilibrium in community. Then we have the ecosystem engineers. So in the structure that these ecosystem engineers create, pores and macro aggregates, you will find an accumulation of the structures coming from lower scales. You see, it's very well known, for example, that an earthworm cast actually is a structure that in which microbial aggregates and microaggregates made by microbial activities are accumulated. And then the next scale is that of a community of engineers in a soil. Now we are in a whole soil horizon. And the next one is ecosystem and landscape. The important thing to realize is that ecosystem services, this function that we expect from the ecosystems to provide us, are produced at the end of this long chain. So there is a simple consequence that may seem very obvious, that if you want to manage soil ecosystem services, you need to manage all the different scales of this machine. And this is how the machine works, you see. So here we have the different clogs. The first thing we've seen is that ecosystem engineers activate and select microorganisms, because ecosystem engineers, earthworms, termites, are not able to digest anything without the help of microbes. You see, this has been very well demonstrated in conclusive experiments. So an earthworm or a termite only digests because microbes do it for it in its gut. But in its gut, there is activation and selection of microorganisms. Microorganisms will release energy for all these engineers, and these engineers will use this energy to build the soil physical structure, that is, build macroaggregates and porosity, but also operate all the dynamics of soil organic matter with its two components, mineralization that release natural fertilizers, and humification that conserve organic matter into the soil. So we get there the final ecosystem services, which is water infiltration and storage, protection for erosion, nutrient cycling, climate regulation. So this is, we like to use this model because it's something you can show to non-scientist people. And I think the analogy with a machine where everyone has its place and where cooperation is the main rule, people understand it very well. Okay, so now let's coming into a bit more of details. We see that, for example, in the physical engineering function, invertebrates will have different kinds of activities. You see, for example, we see that some macroinvertebrates decompact the soil. This is the case of the ants that you see here below, or this is an earthworm that make very fragile and tiny cast. So these ones will create pores in the soil. Other ones will, on the contrary, compact it, produce very compact casts that may have bulk densities of more than 2, which is really a very, very compact. And down in the down part, it's an ant from northern Argentina, you see, Camponotus punctulatus, that is able to build 2,000 of these nests in a single hectare of abandoned rice fields. So we have different kinds of physical activities. And if we come back to this soil thing section where you are still living at the moment, you see that compacting species will produce these compact structures that are like bricks, you see, in the fabric of the soil, and other ones that will create a loose part. And the consequence of that is that water will be able to infiltrate and circulate. So an interesting thing is that people rarely relate the removal of macroinvertebrates from cultivated soils by pesticides with a collapse in the physical structure. It's only because this cast, these earthworm casts or termite buildings are so solid and that they may last years, you see, until they become decomposed. So you may still have a very intensive crop field with grumous structure, but I can tell you that within 5, 10, I don't know how many years, this grumous structure will disappear, and it's something that can be seen. Okay, so this and other examples of physical engineering, you see, it's not only porosity and aggregation, you see. Sometimes you can see very strange things like in this soil from Madagascar, a giant earthworm, you see, these holes are, you see the size of these galleries. They build a drainage system in the deep soil, you see, something really extraordinary. Or in this soil from the oxysol from the Colombian llanos, where you see an earthworm that get down 60 centimeters down in the soil for its annual diapause, leaving a cast rich in organic matter that is used by roots to penetrate the soil and get the nutrients. There are all other kinds of ecosystem engineering that macroinvertebrates do, and this is why we think they have a very important function in soil. We have seen physical engineering, but there is also community engineering, and we see that by creating habitats and favoring some functions, or sometimes by burying litter, they shape the communities of other invertebrates, microbes, and even plants, you see. So this engineering is done through two kinds of processes. The production of biogenic structures, but also ecological mediators. You see, I think it's a thing very important. I think this earthworm mucus is something very, very special, you see, and very little investigation is not on these ecological mediators that may be signaling or energetic molecules. Plant ecologists have advanced a lot in that, but we soil zoologists have not done much. Yeah. So community engineering is very important, and also biochemical engineering, you see. It's also a very promising field of investigation. You know, some years ago, Manuel Blouin found that earthworms can change the expression of genes of plants attacked by nematodes, you see. So the fact that they trigger changes in gene expression is something very strong that should be studied much more and used in agroecological practices, you see. So this is where we are, and so I think this introduction was important, just to convince you that macroinvertebrates, just like microbes or nematodes, are very important elements in the soil, you see. And just say they are not only important because I like them, you see, but I've also found some other reasons for them to be respected, studied, and used. So macroinvertebrates as indicators of soil quality, I think it's quite obvious, not only because all this theoretical development I did, but also because for their diversity and for the taxonomic and functional diversity. Many years ago, well, I lost the count, there was an international project that was called Tropical Soil Biology and Fertility. This was launched by great, mainly British soil ecologists, you see, Mike Swift, John Anderson. And what they wanted to, they had this idea to put ecology in agronomy, you see. Well, at that time, it was a big challenge. Well, it looks like it still is, but not so much. So I entered into this project and wanted to promote soil macroinvertebrate, especially earthworms. But so we proposed a very simple method, you see, which is like just, you know, with this metallic frame, you take a sample of soil, when 25 by 25 centimeters, you take up to 3 layers if you have time and labor, and put people looking for macroinvertebrates. Yeah. So because it's very simple and it very soon gave a very spectacular results, this method has been used by many people. And there are many papers using it. And apart from the fact that it has become a standard ISO method, it's also a very important way for looking at the soil together. You see. I just selected two of these places I think I've been maybe in several hundred places doing that. So you spend the whole day looking for macroinvertebrates like that, you see. And it might seem very boring, repetitive, and time consuming and losing your time. But it is not, you see, because when you do that, well, I don't know how it works, but I think some of the soil science enters into your finger and comes to your brain, you see. Maybe it's not a very scientific explanation, but but you can observe lots of stories, you see, when opening the soil. So this method showed a very high sensitivity since the beginning, and this is the first result we published with this method, a set of land use systems in Peruvian Amazonia. You see where you see that forest. The size of the circle represents proportional to the biomass. So if you have many colors, you have more diversity. So you see that when you transform the forest into high input agriculture, you lose most of the macroinvertebrates. If you put a pasture, you lose lots of macroinvertebrates, but you get lots of this blue thing that is an invasive earthworms that will do the job there, and fallows will give intermediate results. So we got at the time we worked on this. We built a database, and at the time we started to process data with Jérôme Mathieu and many other friends. We had more than 4,000 sites, you see, and all studied with the same method, you see. And this is where they are distributed, you see. There are a couple of big holes, but well, don't worry. They are being now filled with the new part of the project. So more than 4,000 sites with a very wide range of plant cover, altitude, latitude, and soil conditions. So this database is now available at this address and as part of the published paper, you see. So this paper was published this year in Global Ecology and Biogeography. And this is what an average worldwide community would look like, you know. More than 7 individuals per square meter, 5 orders on average, and this is more or less what it looks like with 3 dominant groups. You see ants, termites, and earthworms, and a very interesting structure of the data. Very simple, you see. All our sites will be ordinated this is a PCA analysis, multivariate analysis. So the first main factor will be abundance and diversity. And the second one, dominance of social insects, or as opposed to all the other ones, earthworms, myriapods, etc. So this is how it looks. And this community is sensitive to lots of effects, you see. In this yellow box, you see that plant cover will alone explain 11.8% of the variance, elevation 14%, latitude, temperature, texture. So really, a soil macrofauna community tells you very much about how the soil is. Another important point is that the separation into these very broad groups, you see, is very efficient because you know, if you would go to the species or even family level, Coleoptera, for example, would be totally different. But in the context where we use the data and comparing different types of land use and geographical places, Coleoptera, whatever they are, will more or less have the same function in Amazonia or in Sweden, you see. So this is a very important point that that makes the use of these macroinvertebrates very efficient. So this is well, the main result we have. Unfortunately, we found that lots of these macroinvertebrates are disappear as a consequence of intensive agriculture. Well, this is no news, you see. From tropical forest to tropical annual crops, you lose half of it and a bit more in temperate regions. Another thing is that you have higher densities in hot climates than in cold climates. But maybe the good news is this thing intermediate between annual crops and original systems, that whenever you add trees in to the system, here you have annual crops associated with trees, you can keep a large proportion of this macroinvertebrate community. So trees are very important for macroinvertebrates, and I think it's a very simple and practical conclusion of this work, you see. Well, I will not detail much this. This is how the communities respond to different different attributes. There you will have more macroinvertebrates if you're close to the equator at a temperature between 20 and 30 degrees, with at an altitude between 1,000 and 2,000 meters, with a sandy texture and a rainfall of 2,000 to 2,500 millimeters per year. Now, the next step we've been doing is that, you know, it's nice to see, you see that macroinvertebrates respond. How could we, can we put together all this information and get a single indicator, you see? Because, you know, if you want to be practical, you have to be simple. So we've devoted considerable effort to do that. And actually, it's we have been able to propose synthetic indicators of macroinvertebrate community. So it goes through three steps. I'm not going to detail much. First, it's based on PCA analysis, where you have to compare several sites with different types of land use, and you use some of the outputs of the PCA. You see the weight of the PCA factors, correlations between variables and factors, and coordinates of what. Then you have a formula here, and this formula gives you a raw indicator value, and by homothetic transformation, you can put it, transform it to a 0.1 to 1.0 range. So we've been applying that in many places. This is an example from Colombia, you see, an interesting example that shows how these indicators changes among different types of land use. You have the native savanna here. You see that improved pastures can improve this indicator a bit, but it's not significant. That in the conditions where they are done, oil palm and rubber plantations maintain more or less a good level of community. These systems had no herbicides, I must tell, and annual crops will still have a much lower value. The other thing important is that in the same study, we calculated indicators, synthetic indicators with the same methodology for aggregate morphology, physical variables, you know, porosity, aggregation, carbon in soil and vegetation, chemical variables. And we found out that all these indicators are linked together and to the soil macrofauna indicator. So this is a very important thing. This goes along with this concept of bundles of ecosystem services. Literature now has many examples of studies where people show that if you improve biodiversity, you will improve all kinds of things. You will improve plant production, you will improve soil function, you see. So from here comes the idea to use soil macrofauna as a general indicator, because you've seen that it's not very complicated to get this soil macrofauna analyzed, you see, and even identified, because you know, most people, especially farmers, they know what diplopod is. They can separate different types of ants and worms. So it's, you know, instead of measuring things, complex things like carbon or soil porosity or aggregation, if it would be enough to look at the macrofauna community, it would be a great advance. No? So these are examples of the different communities where here you have different types of land use. I'm not going to detail. And here you have the PCA results of the PCA analysis. And you see that all these analyses show the same results. You see that the community, the different types of land use are separated by the abundance, density, and the taxonomic richness of the different groups. And you have a pole here with more social insights and other things there. So we did that for, it's a student called Eliana Hurtado, who is finishing her master degree on that. And she found out that, you see, I gave her, she was taking many places. You see here, you've got places from Amazonia, here tea garden plantations from China, French forest. So we came to the conclusion that could we design a global indicator that would be valid everywhere? And the answer is yes, you see. So this is just some of the things she did. We have a general global indicator, and we calculate this indicator for the place here, and calculate the correlation between the data got with the global indicator and with the local indicator. And you see that correlations are very high. So I think we are going to publish very soon the magical formula that will allow us to evaluate value of a macroinvertebrate communities wherever you are, you see. So now, just finishing with some practical issues, you see, what can we do with this research? I think the first important thing I have mentioned is to provide the farmer with a tool to evaluate his ecosystems, the ecosystem services produced at the farms. Because we are in a very nasty situation where the farmer is doing lots of things, you see. Is really maintaining life, fertility, water, climate, and people education in his farm, and is only paid for the crop, which is sometimes very unpredictable, and at a price that is many times does not represent the value of the work and knowledge that he has put. So I think this situation cannot be accepted anymore, and we scientists have to provide the farmer with tools to measure, to quantify the work he is doing and the services is doing for the society now. So we have started with finding indicator species. There is a method which is called INVAL that allow to relate species or groups to an attribute of the soil that is defined with our indicators. And here, for example, in this paper by Sanabria, published a few years ago, we can see that some ants, when they are present, indicate high levels of soil biodiversity. So the next step was to work with the farmers. And we came to the farmers with two things, you see. The scientist comes with a list of species that are indicators of different things. In this table, what you have actually is ecosystem services versus different soil macroinvertebrates. And we have the scientific knowledge, and we will confront it with the farmer's knowledge, you see. So for example, we ask a farmer if he finds a chilopoda, can he relate it with high soil, high, medium, or low soil fertility? So we have our scientific answer, and the farmer will give his one. So in this preliminary work, you can see that 60% of the cases in red, we had opposed perceptions, but 1/3 in green showed the similar evaluation, and yellow were just compatible. So this was a preliminary study, and that we want to continue, and because the real issue is to get normative systems, you see. So the next steps are the first one is to keep on collecting and analyzing data. The guy here, the person to contact is Jérôme Mathieu at the University of Paris, who is running a project with IDIV in Germany called the Global Soil Macrofauna Consortium. And the idea is to collect, to put all the available data on the database and keep on analyzing them. Second thing is how can we use macrofauna as a resource to manage in agroecological practices. So just two ideas. One, we have a specific technology called FBO, like fertilisation bioorganique, that was once patented. But well, you can get it at no cost, no problem. And it's a nucleation technique that uses a mixture of different types of organic matter and earthworms in trenches. You see, we have applied it in many situations, from tea garden plantations in China and India to sites here in Colombia. And it works very well. The only problem, if it's a problem, is that it costs $1,500 for each hectare to do this kind of restoration. So if you don't have public policies behind that to help the farmer, he will not do it. The other thing on the left is a more general approach. Restoring natural capital. in this figure, you have this model of self-organization, indicating all the things you can do. You see, from putting fuel into the machine, that is organic matter. You see, any restoration of soil starts with bringing organic matter in whatever form you can, and then manipulating microorganisms, manipulating macroinvertebrates if you can, directly or indirectly. The use of companion plants, that is very important for example, in fixing nitrogen or fighting pests, you see, and also creation of sustainable landscapes. You see, I think now lots of research must be done at the scale of the landscapes, not only at the scale of plots. Third is work with farmers to develop normative systems of evaluation of soil-based ecosystem services with macrofauna. And I can tell you it's a very pleasant exercise, you see, because many farmers, for the kind of society we're living on, have a very low self-estimate, you see. And it's a pity because, farmers are very strong and knowledgeable people in many occasions, even if they are poor, you see. And you can do that with children, and children love to do that, you see. So, and finally, just in relation with the issue of self-estimate of farmers, develop social capital in rural areas. I think it's not only a matter of being sorry of the poor conditions of the farmers, see. The problem is more strategic, you see, because if you don't get happy farmers in the rural areas, who will produce our food and in which conditions? On the contrary, you see, agroecology, it may provide lots of very interesting jobs, you see, for people who will like to live in rural areas. So I think it's very important, and I must confess this has become maybe my main activity. And I invite you to visit the environmental education program that I'm developing with my musicians. I have a professional band. I am the only no professional, but they are very kind with me. And this band is called Hijos de Gaia. You can find in YouTube all our songs and videos. And well, most of them are in Spanish or French, but several of them also have English subtitles. Well, thank you very much. And if you have any questions or songs to sing, I'm there to listen.
Patrick, thank you very much for a wonderful lecture. It's great seeing how you've taken this from the biology of, just how the earthworms and the macrofauna live, all the way up to how this information becomes practical in a global sense as indicators. So I know you've got a lot of questions. I've seen them over here popping up, but I want to thank you again for the lecture. And then if you would like to look at these, this is in the question and answer, everyone. If you have questions for Patrick, it goes in the question and answer. And we start off with the top one. Can you see those, Patrick? Have you found that yet?
Yes.
Okay, great. If you want to go ahead and answer and just go down at your own pace.
So I can start with the first one.
You can stop on the screen. Yeah, even though we like your YouTube band.
From Maulana. How important is soil fauna in the agricultural ecosystems? Yeah, since many of the roles get replaced with the land management. I think it's I think I have answered this question. You see, the problem is that all the land industrial, let's say, conventional land management options don't do a good job, you see. tillage destroys soil physical structures and finishes with erosion. Chemical fertilization has a negative impact on water quality because because fertilizers are not only partly used by plants, and pesticides kill lots of non-target organisms. So I think as much as we can replace these functions by macroinvertebrates, it's better. I'm not extremist in that view, and I consider that you can have lots of intermediate systems between the totally agroecological and totally conventional, you see. But it's not acceptable now to see bare soils with no organic inputs, no plant cover. Could you hear me?
Yes, I can.
Okay.
Yeah.
So
yeah.
I think now the second one, which organisms create compaction? Yeah, okay. This is an interesting issue because the only paper we have published in Nature was to blaming earthworms for compacting the soil in Amazonia to a point that nothing would grow, you see. So I think I showed that in the slide where you have the compacting and decompacting species. It's a matter of biodiversity, of equilibrium among ecological functions. So the most biodiversity you have, the less risk you will have to get compaction. But in my experience, compaction is very rare, and the most important cases of compaction I've seen is by cows, you see, which you cannot blame worms for for compacting soil in pastures. The third one, yeah. Okay, something about ecological categories. Yeah, I think it's important to get the macroinvertebrates classified into categories. I've shown at the beginning a very raw classification between ecosystem engineers and then inside ecosystem engineers, compacting and decompacting. You must know that there is a very active debate on earthworms to define ecological categories. I think it has to be done. We don't have such a thing so much developed for ants and termites. I think the issue would be to define categories in terms of global soil function. You see, what these organisms do to the soil, and maybe classify them according to their contribution to the three types of ecosystem engineering I've mentioned: physical, community, and biochemical engineering. This would be my recommendation.
Hey, Patrick, will you read the questions so that we make sure we get them on the tape?
Yeah, the question was, Patrick, there is an increasing number of studies aiming to define functional groups for soil microinvertebrates. What is your opinion on that? So the next one is from Geneviève Stephens. I've studied in soils in steppe grasslands of Kazakhstan, relatively undisturbed or grazed, but never plowed. I didn't see a single macroinvertebrate after weeks of sampling. Could you explain what this is? Sorry, I cannot explain. Please invite me there. I will go and see. No, seriously, I think there may be very strong restriction to life of macroinvertebrates there, and maybe the fauna that is doing the job will be microarthropods and and microfauna. You see, maybe we are in a situation where conditions are bad. But still, I don't know, because I've been sampling in places like Kuwait or Saudi Arabia, which are very dry. And I would always find some coleoptera and ants. So maybe Geneviève, we could have a private talk too, because I'm quite puzzled by your question, and it needs maybe a more thorough discussion. Tobias is asking, is it correctly understood that the standardized sampling relies on counting the individual of different major taxa in a standard size soil sample? Yes, I think it is the key point. You see, you have to do a quantitative sampling just to be sure you will collect everything. Because if you only do a qualitative sampling, you may miss some small things. You see. Next one, Tobias. Are results of this standard sample deposited in a public accessible repository? I say yes. If you get the papers that we publish in Global Ecology and Biogeography, there is a reference of the repository, and you can also make contact with Jérôme Mathieu, who is now managing the wolfing. Calculate the indicator. I think it's also, there is a paper on that by Velasquez et al. 2007, but you can contact me and I can give maybe more detailed information on that. Michael O'Brien, with a beautiful, with a nice compliment. Thank you very much. I would like to know your opinion on experience with vermicomposting and how it rolls and scope. Yeah, okay. I'm not a specialist of vermicomposting, except that I'm giving classes of formation of vermicompost in colleges, because I think everyone should have a vermicompost process at home. You see, because well, it's a general civilization problem. No, I think vermicompost is the best compost you can get. You see, it's so strong that the liquid you get, you have to dilute it 10 times before putting to a plant if you don't want to kill the plant. So I think that I recommend very much the use of it. Andres Felipe, how significant the production of nitrous oxide by the microbiome earthworm interaction? There is a whole set of research about earthworms emitting nitrous oxide to the atmosphere. I fighted a lot against these views, because these are things that occur in a specific laboratory experiments. You see, where you put too many worms with too much water, you create conditions for release of nitrous oxide. But although it can happen in nature, because nitrous oxide has always been part of the ozone cycle, you see, I don't think it's a reason why not to have earthworms in agricultural field. Armand Richard asks, is it possible to increase the number of invertebrates in farms by just planting trees? The response is yes, you see. I think anything that will bring increases in amount and diversity of organic input will have a positive effect on soil macro and invertebrates. Carlos, hola Carlos, buenos días. Do you think megaphone should be incorporated in your theoretical approach? Of course, they should be in places where they are important. You see, yes. it's not very important. It's not to be seen in many soils, but they exist, and also they may have effects at a much larger time scales. Yinon, what do you think of total biomass per square meter as a simple indicator? I think it's a bit too rough, you see. You know, at the beginning, we were measuring biomass in addition to densities. And finally, we gave up using biomass because it gives less, it's less discriminant than density, and it is very much work. So I think density is much better. Widjat Mani, there any standard of macrofauna density to good soil quality? Yeah, I think we are going to provide this information with the global indicators that will be soon published. Anonymous, thank you. Well, thank you, Anonymous. Carlos, I meant the megaphone. Yeah, okay. I answered Carlos's question. Can you please inform me about the openness of data? Yes, I think it's. I told it. It's on a free access. You see. Tati, is there anything specific sampling methods to use the synthetic? Yeah, I think what you do is applying the ISO TSBF sampling method. And then you use a number of the different invertebrates you found in the formula. So you will get an evaluation from 0.1 to 1.0, and it will give you if your how your soil compares with the 4,000 other soils of the database, and if you have different types of land use that you have sampled, it will tell you which one is best.
Aha. You got through those very quickly. Are there any more questions that people would like to ask? Okay, Patrick, do you have any further comments? I mean, I think the biggest thing is people are really interested in where to get the data.
Yeah.
And then also how it applies to their particular, their land where they are and how useful is that open. So I think that's really important.
Yeah, I think the methodology is very simple, and it has no cost, you see. And students like to do it because they like to see fauna, to see soil, and doing things in the field, you see. I have two comments, you see. One for you, Diana.
Yes.
The comment for you is that we should investigate more the relationship between all these micro food web invertebrates and ecosystem engineers, you see. Mm-hmm. And I always have a very tough question for people is what happens when an earthworm ingests the soil food web, you see? Mm-hmm. Because soil food web is very well defined, and it's a very sophisticated structure. But we know that the worms every year the soil in which the food web is contained goes through an earthworm gut or is in contact with a root or a termite. So I think we have a very lack of investigation on that, you see. And no, the other comment is really that we need to change our way of working, work with farmers, you see, be more social in our approaches, go to the farms, work with farmers, and also try and make our communication a bit more fancy, you see. I don't know if my songs are the way to do it. There may be lots of ways to do it, but we cannot keep on being confined to to our high quality scientific publications that are very important, but it's not enough.
Mhm.
Ah,
Yeah, I
she,
think you've
she,
shown
I
this,
want-
and now we have 2 more questions.
yeah, she from China. It was her birthday 2 days ago, so happy birthday, Chi.
Yeah.
Yes,
Read the
I
question.
don't.
Yeah.
Yes, Eisenia foetida is a composting worm, so I don't think it's the suitable species for the FBO technology that requires endogeic soil feeding earthworms. But if you can demonstrate me the opposite, I will be very pleased to see it. You see, because I know that in China, lots of people have developed the FBO technology with the kind of worms they had, that is Eisenia foetida. So I guess Chi has the answer. Tobias, yes, the method is publicly available, and if you have problems to get it, you just write to me, and I will be pleased to give all the details. And Michael, artificially increase the quantum of worm? No, I think if you bring worms and there's not enough food and space to host them, they will just die. So increase earthworms is by improving the general condition for the life. And even the FBO technology, you bring worms, but you organize in small trenches the best possible condition for the life.
I have a question that I can verbally ask you is what is the status of macrofauna and as invasive species? Is that
Okay.
growing since you started studying that you're seeing more species where they don't belong, or do we have a good idea of what are endemic species and what are not?
Very interesting question. I can reply for earthworms, you see.
Okay.
All the cultivated fields all over the earth, with a few exceptions, are invaded by species that have very broad ranges of distribution, that are very tolerant to any kind of nasty conditions, and they are generally parthenogenic.
Mm.
So the risk, what happens is that agriculture is uniformizing earthworm communities tremendously. You see, you have an example of that in Northern America, where the most efficient lombricid earthworms are entering. In the tropics, we have a few species. It's interesting. You can have a community with an American and an Asiatic and an African earthworm together. You see, but the problem is that the great part of the native biodiversity disappears. Ants, I think, but I may be wrong. There are not many invasive ant species apart from the fire ant species. Invasive macroinvertebrate species are not so far considered as a problem, except that invasive earthworm at least are eliminating the local biodiversity.
Okay. I think we'll take this one last question. And I agree with Carlos on the comment, excellent presentation. So let's go to the question, the very last question.
Carlos is a good friend, but I was not the one who asked him to put this. Okay. This question is very important too, and I have no answer to that, you see. Because in the literature,
read
you
the question,
can...
read the question, yeah. Read the question. We have to have it on tape.
Sorry.
Yes.
Is it possible to isolate beneficial or specific function of gut microorganisms of earthworms to produce biofertilizer? I have no answer, but it's maybe because I'm very short of knowledge on this topic. But I think it's a very important question because I always question the value and of all these microbial biofertilizers, you see. Because most of the time, since they are protected by industrial secret, they are not, they may not be as reliable as people pretend. So I think the question of Brian is very important, and it's important to do in research on that.
Okay, well, everyone, I know we're all clapping around the world and thanking Patrick for his lecture today. Patrick, it's great to see you. Keep doing the good work, and we'll see you later. Goodbye, everyone. Thanks so much.
Bye-bye, everybody. And I was very pleased to be able to give this lecture, you see, because it's, well, many years of working with soils, with farmers, with scientists, and it's good to be able to share things that like that, you see, that not always go in publications.
That's right.
So a big hug and kiss to everybody, and let's keep on working on this very important topic. And whatever information or help you need from me, I will be very, very pleased to provide.
Okay, thanks so much. Bye, everyone.
Bye-bye.